Thank you, Pedro, and thanks for asking me to speak at this meeting. I was at the first one, and that was a big influence and impact on my career, and especially pleased to speak since this is in honor of Emery, who was a very special person, was always very kind to me. I'm going to talk about the phylogenomics of the Dothideomycetes, a project we've had ongoing with researchers here at CBS and also at the Joint Genome Institute in Walnut Creek, California. And here are some of the people involved at JGI: Igor Grigoriev and Sajid Haradas, and here at CBS, Manfred Bender and Pedro Kraus. So a little background: this genomics effort in the Dothideomycetes started back in the mid-2000s and building a community, and it was really led by these three people, Steve Goodwin, Gert Hema, and Gillian Turgeon. And they started with the kind of sequencing their three pet organisms, the fungi they really spent a lot of time on research and working, and slowly started building by adding additional genomes, mainly around plant pathology and how that related to the Joint Genome Institute's mission of biofuels and feedstocks and whatnot. And they started forming these groups that they called jamborees or workshops, and they were held sometimes at JGI, which is in Walnut Creek, California, near San Francisco, and then at the Fungal Genetics Conference, which is at Asilomar, just south of there in Monterey. And it started out as a small group, and now it's grown to a large group where it's about 50 to 75 people at each meeting, depending on the group. And what I'm going to talk about is what happened here at the beginning of 2013, mid-2013, with the beginning of the 1,000 Fungal Genomes Project, which was really leveraged by the Dothideomycete community. So if you go to this URL, here you can find out information about different groups of fungi and genomes that have been sequenced. And you look at the Dothideomycetes, and we are now up to 91 genomes that have been sequenced across the diversity of the class. 29 of those have been published. The majority of them have not been published yet, but have just gone through production in QC. And even though this is a large number of genomes for a particular group, it's still about a third of the higher level diversity when we think about families or orders. And I really want to call out here the group at CBS, Manfred Bender, who was here previously, and Pedro Kraus, the hardest working man in mycology, for their effort. Of these genomes, this group has produced about 50 of these 91 genomes. This is having a tremendous impact on the Dothideomycete community on a global scale. This is just, many of you have seen this, so this will be kind of my last shameless plug for the 1,000 Fungal Genomes Project. But this sequencing occurred as part of this, and Jason Steisch is the co-PI of this project, and Igor Grigoriev is our JGI leader who's responsible for kind of organizing on that end. And the goal of this project is to facilitate sequencing across the kingdom to really cover phylogenetic diversity. And in doing so, produce a genome-scale phylogeny for the kingdom and to test and refine hypotheses about the evolution of fungal genomes. And to think about this as a pan-fungal genome and to move a lot of science, or not necessarily from model systems, but to move technologies and advancements from model systems into non-model systems, into the phylogenetic diversity of fungi. So you can learn more about this project here at 1000fungalgenomes.org. Our sampling is phylogenetic. We really want to sample the diversity. You know, the pathogens are going to get sampled, the human pathogens are going to get sampled, but all of these evolve from what we recognize as the phylogenetic diversity of fungi. And it's important to capture that so we can really understand a lot of these processes and patterns. Our goal is to sequence a minimum of 2 species for every family level clade and focusing on these undersampled lineages. And I really want to highlight that right now we have what's called a grad student or postdoc challenge, that graduate students and postdocs, we want you to take full advantage and exploit this program to have data collected for your project, for your research. And it's free. It doesn't cost you anything. All right, so our project in 1,000 fungal genomes, we're up to about 300 genomes, closing in on that. I'm showing you a tree of 240 of them, with another 60 that are about in QC and coming into this. This tree right here was produced from an alignment of 180 proteins, about 75,000 amino acids. And you can see the major groups here. It's heavily sampled for the dicaria with a lighter sampling of the early diverging fungi, something that we want to correct over the next year or two. So this is obviously hard to read, and so we're going to simplify that. The Ascomycota, a lot of the topology that Thorsten was talking about. The Tephrinomycotina is the earliest diverging lineage. The Saccharomycotina and the Pizizomycotina sister groups. And within that, we have the earliest diverging branches of Pizizomycetes and Orbiliomycetes, and then the Leiotiomyceta. And our focus here is on the Dothideomycetes, which forms a monophyletic group with the Eurotiomycetes, Lecanoromycetes, and Zalanomycetes. So there's really no name in our taxonomic rank to call this unless we go with a rankless approach, but I won't go into detail on that. So what I'm going to focus on now is give you just a little bit of the overview of the topology within the Dothideomycetes, very briefly, and then I'm going to focus on the genomic evolution, what we're kind of beginning to learn from these data and these analyses. So this is a little bit expanded sampling. This is from 300 proteins. It's a RAxML analysis, and I'm showing here the major orders, or the orders that are present, where they exist or where they have names. And there are two major spheres or groups of the Dothideomycetes, and these correspond roughly to the Pelosporomycetidae and the Dothideomycetidae from Conrad Schoch's paper and classification in 2009. And at that time, those were multi-gene trees, 4, 5, 6 genes, and the resolution of these earlier diverging nodes was problematic, where this root was. And with these analyses, we're able to confidently divide this or place this root into these two major groups that potentially we could just recognize as two major subclasses of the Dothideomycetes, the Pelosporomycetidae and the Dothideomycetidae. So I'm going to look at each of these in a little bit more detail. Within the Pleosporales, we've sampled 23 families. I say we, I want to emphasize against the Dothideomycetes community. Roughly half of these were sampled through the efforts of CBS and the 1,000 Fungal Genomes Project, and the rest were through other researchers. But these are all the data that are available on the JGI site. So we've sampled 23 families from the Pleosporales, which is about half of the families we recognize today. And there's been the ability to place some things within the Pleosporales that were in Cetus, like this Eosphaeria arxii. And there are some families in this initial sampling that are turning out to not be monophyletic and will have to be addressed. Some major findings from this. Placing Cenococcum in a more defined group, in a more explicit group. This is in collaboration with Francis Martin, who does a lot of work in mycorrhizal genomics. And Cenococcum places here in the middle lineales. And we knew that it went with Glonium, shown here, which is a saprobic hysteriate Dothideomycete. And here's Cenococcum on a root tip. It's probably the most common ectomycorrhizal Ascomycete in nature. We don't know, we've never seen a sexual reproduction of this, but we know it's placed within the Dothideomycetes. And now we can place it in greater context with things like Lepidopterella, which is an aquatic Discomycete. So a fair amount of ecological diversity in these clades as well. Pseudovergaria, which is a hyperparasite of rust. Again, sexual state's not known. It was provisionally placed in the Capnodiales. This says no, not the Capnodiales, but in the Pelosporomycetidae, perhaps on its own lineage. The Botrosphaeriales, which includes a number of important plant pathogens, causing cankers and different types of tree pathogens. The Patalariales, which is an order of primarily saprobic fungi. The Venturiales, which is best known for the apple scab here in Venturia. And then an example from the Microtheriales is Allographum, which is a saprobic organism. And just very briefly, in the Dothideomycetidae, most of our sampling has been in the Capnodiales for obvious reasons associated with plant pathology, where you have Spirulina, Zymoseptoria. But in here we find interesting ecologies like Zasmidium, which is the wine cellar fungus, the angel share fungus, which is able to metabolize volatiles. Interestingly, in here we have the Stratosphaeriaceae, including things like Stratospherium baldonia, which also can presumably metabolize volatiles. In here we see Podrea, which is on human hair. And it's an oddball organism placed in amongst these, what some people have called extremophiles. And we're going to come back to that. And then Capnodiaceae and some small examples from Dothideales, Myriangiales, and the one lichened example here in Tryptothelium, which is done by a group led by Francois Luzzoni, Daniele Armileo, and Stephen Goodwin. So, we have this large-scale phylogeny. And one of the things we wanted to look at are other sources of information from the data that support these relationships. And one of the things we looked at was synteny, which is just simply the occurrence of genes in the same order between two genomes. And as this goes from light to dark, you have greater levels of synteny. So here we see Zopfia, which is placed here in the Pleosporales. As you can see, across closely related organisms, it shares a lot of synteny. Pseudovigaria, that strange parasite, hyperparasite of rust, here in its synteny, it's placed as well in this tree, but it's not really in a strong block like this. And then Podrea, the parasite of human hair that's placed in the Stratospheriaceae. Here you see it in a lighter block, but still some synteny with its close relatives. And I'm going to come back to this point again in this Podrea. All right, so a little bit of information using this tree to look at some traits about genomic evolution. One of the first things we'll talk about is just size of these genomes. They average about 36, 37 megabases in length. And here I'm showing in the dark is the non-repetitive elements of the genome. Most of this is gene content, but not all, but it's non-repetitive. And then where you see in the light gray is repetitive, transposable elements and things of that nature. And what you see is even though there's about 40 megabases is the average, we have two examples here that are quite large at over 150 megabases. And these include the Zopfia, which is we placed in the Pleosporales, and Cenococcum, which is that ectomycorrhizal fungus, the most common ectomycorrhizal ascomycete. And you can see in both of these cases, the expansion of these genomes is mainly, not entirely, but mainly from non-coding or repetitive elements in the genome. On the other end of this, there's that Podrea, highly reduced, the smallest genome at about 17 megabases. So this is one of the smallest genomes of filamentous ascomycetes. Highly reduced, but look at this branch. It's on a very long branch. So this genome is contracted, and we're seeing higher rates of nucleotide substitution, mutations at the coding level, resulting in amino acid changes. Now, when we look at number of genes in these systems, the average gene count's about 13,000 genes, and it varies from as little as 7,500 to a max of 21,000. And what you're seeing here in blue are genes that are found in core fungal genomes, core fungal genes. So when we compare these across all fungal genomes, a data set of about 450, these are conserved genes that we find throughout fungi. This next group in this maroon or red are genes that are unique to the Dicaria, and green unique to the Ascomycota, and purple unique to the Dothideomycetes, and then in orange at the tips unique to those organisms, those species. Now, here's that Zophia, which is one of our largest genomes, and it has a larger gene count. And it looks like there have been multiple segmental duplications in this genome that have resulted in higher rates or larger numbers of genes. But if you look at Coccum, even though it has the largest genome of any of these, it's 170 megabases, this unique ecology of being an ectomycorrhizal fungus for the Dothideomycetes, its gene content looks very similar to its relatives. And then here we are with Prodrea again, which is contracted. It's the smallest genome, and it has the fewest number of genes. It's really streamlined its genome associated presumably with its ecology of this shift from plant-associated to being associated with human hair. So if you think back at that synteny, where it had that lighter synteny organization with its close relatives, still present but lighter, a big reason for that is it's lost a lot of those genes. It's simply stripped down. When you look across those, again, if you think about this, about half the genes in your average Dothideomycete genome are core fungal genes. The rest of these, the Dicaria, the Ascomycete specific, the Pizizomycotina specific, the Dothideomycete specific, and then those unique, these each represent about 10% and collectively about half of the genome. So again, for most of these organisms, they're fungal, conserved fungal genes, and what's happening out here at the tips is change associated with these unique genes. All right, and that's what I want to just kind of end up with and touch on briefly. So what you're seeing here are a set of Pearson correlation coefficient analyses where across the x-axis is simply the number of genes. So it goes from 8,000, our smallest, to about 22,000, our largest. And on the y-axis are the number of genes from particular annotations. So here's proteases, this is transporters, these are small secreted proteins, and these are secondary core genes associated with secondary metabolism. And what you see is a general linear correlation. The more genes you have, the more of these genes in these classes that you have. But if you look at these correlations, first of all, none of them have a good correlation here with genome size. As you would expect, genome size is mainly a function of non-coding elements when you increase. So this is the worse, the closer you get to 0, the worse, the closer you get to 1, the better. But what's unique or what's interesting to point out is that the coefficient, this Pearson coefficient or correlation of secondary metabolism and small secreted proteins is quite poor. So the number of genes is not a very good predictor of being particularly rich in small secreted proteins or being particularly rich in secondary metabolism. And these are genes that are associated, we think they're generally associated with the ecology of these organisms and the interactions with other organisms, whether you're pathogen, whether you're interacting with other microbes. So if you look at that at another level, here's that tree again, and what we've shown here are the number of genes in the core genes associated with these pathways, these secondary metabolism pathways. So the first group, this brown are polyketide synthases, the yellow are enzymes that are PKS-like, the orange are non-ribosomal peptide synthetases, and the purple are NRPS-like. And then we have hybrids between those in green, and terpene cyclases in blue, and DMATs in red. And what you see is not a strong correlation with phylogeny. So the increase or decrease in a particular genome associated with secondary metabolism is not an ancestral trait of that group. Rather, it's something that's evolving very rapidly and is more species-specific or pushed at the tips of the tree. Whoop. I thought I had another slide there. I guess I don't. So just to summarize, the phylogenomic analyses support this two-subclass classification of the Dothideomycetes and an expanded view of the Pleiosporomycetidae and the Dothideomycetidae. We're able to place some taxonomically difficult taxa and learned a lot about the evolution of ecologies. So Cenococcum is the only ectomycorrhizal Dothideomycete. It's the most common ectomycorrhiza in natural ecosystems. Pseudovigaria, this hyperparasite of rust, and Prodrea, this organism that has shifted from plant-associated and extremophiles to being associated with human hair and going through this really drastic reduction in its genome. Synteny can provide some support sometimes, not all the time, but sometimes. And genome size and repeat content varies widely and correlates poorly with phylogeny. And the big example of this is that variation in secondary metabolism and small secreted proteins. These genes that we think really function in the ecology of the organisms, especially interactions with other microorganisms and plants, they're organism specific. There's not a lot of correlation at this scale across major clades that we would recognize as orders. And I just want to end again by highlighting collaborators on this, and particularly the group at JGI and the group here at CBS. Thanks a lot.
The floor is open for some questions to Joey.
Could Zopfia be an allopolyploid somewhere in its past?
I don't know. That's definitely a possibility. It looks like right now our best guess is that there are multiple duplications in the genome, but it's not genome-wide. So that could be a mechanism where there's been some type of duplication that could have been genome-wide and then many things that have been lost. So that's possible. We're just kind of scratching the surface of that. So.
On your last point about variation in secondary metabolism is organism-specific, is it possible that it's species-specific rather than organism-specific?
The reason I have organism-specific rather than species-specific is that when you look at multiple isolates within a good species, you see a fair amount of variation as well. And so, that's a relative. There are going to be some secondary metabolites that you find across all of these. They're very conserved.